As we all know it, messenger RNA carries a gene's protein building instructions as a string of three nucleotide codons. Transfer RNA and ribosomal RNA translate those instructions into a protein. What we aren't aware of is that there are recent advances and breakthroughs in this field of study. One of which is that scientists have discovered a new protein that likely will advance the search for new natural antibiotics, according to a study by Texas A&M AgriLife Research. For the past decades, science gave us breakthroughs which led to a fascinating world we are living right now. And for years, science continues to surprise us with recent discoveries which may help us shape our future. Science plays a big factor in our everyday lives. By the way we eat, walk, sleep, and everything we do, each of it is connected to science. Because of science, we discovered things we never expect to find. Now, one of the recent discoveries in science is the long operon associated protein, or LOAP, and the other is the discovery that the protein is frequently found next to the gene clusters that are responsible for producing antibiotics. Scientists know a great deal about proteins that control how certain gene clusters get their start, referred to as transcription initiation, but much less are known about transcription elongation, where proteins keep gene expression going through roadblocks in the DNA sequence. These long chains of genes raise challenges for the molecular machines that decode DNA. Sometimes the molecular machines hit roadblocks called terminators and they stop and fall off the DNA. The LOAP protein is called a processive anti-terminator because it helps the machines stay on DNA and move through the roadblock terminators. Hence, knowing how LOAP works and its prime location could lead scientists to a shortcut for antibiotic production. Sounds wonderful, right? Another discovery is that did you know that heart and brain are very sensitive to mitochondrial defects in early development? According to the findings of the study of the European Journal of Human Genetics, mutations in genes involved in the production of mitochondrial proteins may affect the heart and brain severely during early development and lead to a poor prognosis. The study further highlights the importance of mitochondrial proteins in human health. Identifying specific mutations may help researchers find new ways to treat mitochondrial diseases. Now let's learn what oxidative phosphorylation is. Researchers have searched for genetic defects in 3 children who had severe cardiomyopathy and early onset brain disease. These children also were found to have issues with a process called oxidative phosphorylation. Oxidative phosphorylation is a mitochondrial process through which cells get most of their energy. It is conducted by the oxidative phosphorylation system, which consists of 5 different enzyme complexes in the mitochondria. DNA provides the instructions to make a molecule called RNA. In return, RNA provides instructions to make a protein in a process called translation. On the other hand, mitochondria have its own set of DNAs, which provides instruction for making some of the proteins involved in the oxidative phosphorylation complex. The rest of the instructions come from nuclear DNA, which is located outside the mitochondria in a different cellular compartment called the nucleus. In this way, disorders affecting oxidative phosphorylation can have a dual genetic origin. In order to find out the mutations that cause this kind of disease, process called whole exome sequencing, or WES, is done. Going back to the three children mentioned earlier who had issues on oxidative phosphorylation, researchers used the WES process. In the first patient, researchers identified mutations in the QRSL1 gene, which provides instructions to make a vital mitochondrial protein. In further experiments, researchers were able to restore OS function by using the correct version of the QRSL1 gene. In the second patient, researchers identified two new disease-causing mutations, one from the mother and one from the father, that affected one gene, MT01. In the last patient, researchers again discovered two mutations that affect one gene, AARS2. All of these disease-causing genes discovered through WES are involved in the process of translation of mitochondrial proteins. This leads the study authors to believe that the heart and brain are particularly sensitive to the effects in mitochondrial protein synthesis during late embryonic or early postnatal development. Why? Probably because of the massive mitochondrial biogenesis occurring at that stage. If both the heart and brain are involved, the prognosis is poor with a likely fatal outcome at young age.